I wanted to also highlight that Martin, I like to say the German way, Martin actually was recipient of the 2021 Young Investigator Award. So, you know, it's great to see you a few years later here again, still involved with US HUPO. So I love that. And yeah, thank you very much for your talk. I really enjoyed this. Thanks, Martin.
Thank you.
Okay, great. Well, this brings me to our next speaker. So I'm super happy to have Dr. Yansheng Liu here. And, you know, you had the honor to win the Cotter Award this year at our conference in Chicago. This is a new investigator award that we provide to young, outstanding scientists. And, you know, I think that was a very well-deserved award for you. And, you know, I'm really happy to introduce you, your assistant professor, and now you are actually at Yale University School of Medicine at the Department of Pharmacology. But I also know that in your talk, you talk a little bit about your career path, so I will not say too much here. But yeah, it's our pleasure to have you here today, and you will tell us a bit about the endurance of proteins in context. And Yansheng, thanks very much for being here and spending some time with us today.
Thank you, Birgit. Let me see if we share the screen correctly, or let me try quickly. Mm-hmm. Okay, do you see the screen correctly?
That's great, thank you.
Okay, hello everyone. So I would like to thank US HUPO again for giving this Rob J. Cotter Award to me this year, and Birgit again for introduction. So, I mean, the goal of my award lecture was to share some of our recent studies, but also my personal journey as a postdoc and then a PI. So I had to cut down some slides due to the time limits today, but I still hope I could fulfill this goal. So I was born in the city of Wuhu in Anhui province. Then to broaden my views, I went to Beijing and Shanghai to do my bachelor and PhD, two biggest cities in China. The interaction between proteomics and my life started as early as 2005 when I was a graduate student. After spending 6 years in Shanghai for my PhD, I then moved to Zurich, Switzerland, and carried out my postdoc with Rudy Episode for almost 6 years. And then I moved to Yale and accepted this assistant professor position and started my own lab at the end of 2017. So I met Rudy in the HUPO conference 2010. Here you can see an old picture where Rudy and Rob were talking about the SRM initiative. And I was lucky enough to meet Rudy several times in that HUPO, including a roundtable discussion named the Meeting of the Minds organized by Angeland. I finally had the courage to ask Rudy a few questions. For example, how can we handle the FDR issue in SRM dataset? So a year later, after I arrived at Zurich, some lab members told me something interesting. So they mentioned that Rudy got quite excited after the HUPO trip and said to a few of them that he had met a Chinese student who started to interrogate him about his papers since 1990s, and he had to make up some answers. So I find that's very funny. I also realized that by that time, the lab was actually having this MProfet paper submitted to Nature Method about the same question that I asked Rudy. So I guess that's why I was accepted as a postdoc in the lab, even if I did not have an amazing CV by that time. So my first advice to the young student would be try to ask the correct question and read all the papers. So I want to take this opportunity to thank Rudy as a great mentor for always being extremely supportive during my whole career. My research during the 6 years in the episode group were quite broad and can be summarized into 3 areas. First, the DIA method, or the SWOT version of it. The second is the plasma deglycoproteomics. And the third is investigation of the genetic impact on the human proteome. So because I was there for more than 6 years, I was deeply connected to the lab and also the episode tree, which is a great resource for my research later. Herein, I want to also take the opportunity to sincerely acknowledge this group. Some of them are still my close friends, colleagues, and collaborators these days. So for example, I have been enjoying the collaborations to Hannes Rost and George Rosenberger, two PhD students in bioinformatics by their time in the lab. With respective collaboration, we have generated a prototype software tools such as Trick and IPF for data alignment and PTM analysis in DIA dataset. Ben Collins was another great colleague. Together with Ben and Christy Hunter from SAIX, we were proud to present this multi-laboratory assessment paper on SWOT to the community. So the second advice would be try to be a collaborative lab member. So there is a particular one postdoc study I want to mention here because I succeeded in taking it along as a new research direction in the current lab. This is about the cell aneuploidy. So as we know, the trisomy 21 or the addition of the chromosome 21 is the cause of the human Down syndrome, which is the most popular genetic disease in humans. By that time, we were lucky enough to work with Anton Narakis group in Geneva Medical School and obtained some fibroblast cells from a very interesting isogenic model, which is a monozygotic twin pair discordant for trisomy 21. This means that one of the twins bears an extra copy of chromosome 21, while the other twin's genome is normal. So we can then compare the mRNA expression and also quantify the proteome differences between these twins. Of course, the proteome analysis was based on source mass. And in this plot, you can see that unlike the mRNA level, the reaction of the protein level to the extra chromosome 21 is quite heterogeneous. Basically, if a protein is annotated to participate in a protein complex, and then the upregulation of them will be buffered to 1-to-1. While the protein is outside of a protein complex, it seems the cell failed to do so, and the ratio of upregulation in the trisomy 21 twin is as close as that showed by mRNA, which is 1.5-fold. So then next we measured the protein turnover, and you already heard a lot from Martin about it. But what is interesting is that we immediately confirmed the degradation of the complex yin proteins from chromosome 21 was actually accelerated. So this little bit busy box plot revealed a protein complex stoichiometry controlling mechanism through protein degradation of the complex yin proteins in aneuploid cells. So since then, like Martin, I also get quite fascinated by the concept of protein turnover and lifetime. And this is something that I always think about these days when I meet a new biological question. So in my current independent lab at Yale, there are 3 research focuses. The first is DIA-MS. The second is the role of post-translational modifications in protein turnover and cancer signaling. And the third is the biological variability at different scales. So I will focus on the second topic. So in this research focus, essentially, we want to measure and understand what and how the protein lifetime means to different biological processes and the diseases. So instead of measuring only the protein abundance or the regulation extent, we want to address the question that how long the protein lasts or the active protein lasts, which is a very distinctive question compared to the first one. Furthermore, because post-translational modifications create different proteoforms that are more relevant to protein functions, we want to extend the lifetime measurement to PTM carrying proteins. We refer this concept of measuring the lifetime of the active forms of proteins as the analysis of the protein endurance. So there is already a nice approach in our field measuring the protein turnover or endurance. This is PulseLec that Martin already extensively discussed. We just want to point out that because the PulseLec experiment often involves multiple samples due to the usage of the time course, we reason that we could take the benefit of DIA mass to increase the proteome coverage among these samples, basically to combine PulseSelect and DIA. I will then introduce a few study examples in my lab around this topic. So in the first study at the bioinformatics side, to achieve a better data extraction in PulseSelect DIA, we worked with Spikeonosis and devised a new ISW workflow, the inverted spike-in workflow. ISW workflow was initially used in those SRM experiment in the old days, when the synthesis of the heavy isotopic peptide was quite expensive. Therefore, the workflow was designed for the heavy proteome spiked with a light peptide standard. So this means that in ISW, only the light channel is scored for identification. So this is a perfect match for PulseSelect experiment, because in the early time point, such as 1 hour or 4 hours, the light channel is always there and much more abundant. So we indeed, if we use ISW workflow in PulseSelect DIA dataset, we observed 30% increase of the quantified heavy to light ratios in the early time points. So also because of that we are using DIA, there are actually more data points in this classic PulseSelect experiment, because we now stay at the peptide fragmental level. So we have much more B and Y ions. For quantification, of course, we rely on many Y ions. But if this actually allows us to apply some interference removing algorithm, for example, like the one provided by Spectronaut, to refine the quantitative accuracy. So at the end, the quantification is more robust and have more data points. So we matched our proteomic data with algorithms called RNA splicing isoforms in this study using the RNA-seq data, and then discovered that for the same gene, the protein level lifetime in the absolute scale and the relative scale can be different for different individual alternative spliced forms. Some of these diversities might be functional, for example, for these TPM proteins as reported. So this is about the ISW scoring system. In the second study, at the mass spec side, for a long time, I always wanted to improve the DIA, especially its window settings in Orbitrap machines for Select or PulseSelect application. Here I elaborate why. So as we know in the Select approach, very commonly, we will have the isotope labeled peptide ending with heavy lysine and arginine. And this is similar to PTM, small PTM modifications with a mass deviation of 8 or 10 Dalton. But it's much worse than the small PTMs for analytical chemistry, because the light and the heavy peptide would be always co-eluting from the LC. So if we are using a routine DIA method of large windows, these peptide pairs will very likely to be isolated together in one window and get recorded in the same MS2 spectra. This would make all the B ions cannot be used for select identification and quantification. So we then reason that why don't we use a DIA window with a small window size? If we move to as low as 2.5 mO/Z per window, then all the B ions will be used, can be used for identification and quantification. However, can we directly use such a DIA method with 2.5 Dalton windows? That would be difficult, especially in Orbitrap instrument, and that means more injections and more measurement time. This is not appreciated. That was actually the first reason why DIA window has to be large. So based on this, we therefore designed a new DIA method called Boxcar Max with a goal to achieve small windows with less instrument time. Basically, Boxcar Max is a highly multiplexing method at both MS1 and MS2 levels. At the MS1 level, we used the Boxcar isolation schema published by Matthias Mann's group, but executed that in 4 individual repeated sample injections. And at the MS2 level, we used the multiplexed DIA or 30 MSX scans. So the key is that in each scan, 4 small windows of 2.5 m/z are placed as far as possible between each other. So in this way, the heavy and the light peptide pairs will be analyzed in different MS2 scans. And the BRN suddenly, most of them could be used for select quantification. So we also did not change any DIA data structure. So therefore, all the previous DIA data analysis tools can be directly used for data analysis of Boxcar Max. So we confirmed the quantitative accuracy of Boxcar Max in a standard dilution experiment and a real pulse-lag DIA experiment. In both cases, you can see that Boxcar Max achieved a much higher quantitative accuracy, especially for those extreme ratios. So this is very helpful to calculate the protein lifetime. So in the third study example, we want to apply pulse-lag DIA in an ambitious but basic research question, that is how phosphorylation globally impacts protein turnover. To figure this out, we try to quantify the effect of phosphorylation on protein turnover for a lot of phosphopeptide. So how did we do it? We performed a standard pulse-lag experiment. We used 5% of the peptide digest to perform the routine calculation, as I just told you. But for the 95% of the peptide, we actually performed the peptide enrichment for phosphopeptide, basically a phosphoproteomic analysis. Then we could really match these two data sets with the goal to quantify how would the site-specific protein phosphorylation impact these heavy to light kinetic ratios along the time, and therefore the turnover of the peptide or phosphorylated peptide. So this seems to be a straightforward idea, but nobody did this before this paper, partially because I think because of the high complexity of this protein peptide turnover data. And we had many interesting findings from this pilot data set. Please refer to the paper if you are interested. Just as an example, and you can see from this heat map that the phosphorylation of proteins in certain pathways, such as the translational initiation or the cell cycle, are enriched in the faster end, meaning that their protein lifetime is shorter upon the phosphorylation on particular sites. On the other end, the phosphosite involved in cellular transport processes tend to stabilize their protein expression by phosphorylation. So that's from this heat map, we can see there seems to be a link between phosphosite function and the turnover alteration extent. So this was, of course, based on the classic modeling, calculating the lifetime in the steady state, and was done only for phosphorylation. We're happy to see two follow-up studies, very nice studies from Kuster's lab, extending the observation and analysis to other protein PTMs, such as acetylation and ubiquitination, as well as a recent study from Savatsky's group, refining the calculation models, which seems to be very important for correct biological conclusions. However, all of these are nice, but these are only done in a steady state. How could we really apply such approach in a biological question? Essentially, we want to measure a non-steady state for that. So that's why we recently analyzed a cell starvation model representing a gradual dynamic process. We confirmed that the turnover data of different peptidomes or PTM carrying peptide can be very complex. And in many cases, we could not fit them into the classic model that Martin told you. So this is expected because for the same phosphorylation, it could have distinctive functions in the early or late stages in the biological process. So basically, you cannot simply calculate a lifetime easily for many PTM carrying peptide. We therefore propose that we could convert this curve fitting problem into a time series problem for which the researchers already had a lot of algorithms. This means that for the heavy to light profile of the non-PTM or PTM peptide, we could the PTM carrying peptide could be treated as the experiment condition, while the non-PTM peptide could be treated as the control condition. And then the temporal profile between the control and the treatment conditions can be analyzed by software such as MegaSig Pro to look at their temporal deviating pattern during the this time course. Then we can perform the notation analysis and clustering analysis for those who were filtered to be significant by these algorithms. So this is what we did. We classified 2,000 temporally different phosphosite in the starvation into 6 clusters. Only in cluster 1 and cluster 5, we see this classic pattern. That means the classic delaying and accelerating turnover patterns. For all the other clusters, we actually see quite a lot of complicated patterns. For example, the difference in the first hour of labeling. So we successfully relate some of these patterns and the features and clusters into the phosphorylation stoichiometry. So altogether, we suggest that this simple analysis may facilitate a hypothesis-free understanding about how PTM or phosphorylation dynamically impact protein turnover. So, okay, in the final study, I will quickly mention this unpublished story in the lab, which is about the protein turnover analysis in cancer and euploidy. So here I focused, we focused on the deletion of the chromosome 3P arm and the duplication of the 3Q arm, which are happening in majority of the lung cancer patient, especially lung squamous cell carcinoma. So I unfortunately cannot say more for this study in the webinar due to the time limit. What I can say is that with a very cool collaboration with Alison Taylor in Columbia University, we actually had the chance to access a isogenic cell line model. Essentially, from that model, we could very confidently found that the turnover control is actually different for the gain type or the loss type aneuploidies. For example, why the 3Q gain cells followed the discovered rule of proteomic buffering is exactly as I was telling you about the trisomy 21. In the loss type, however, we did not detect the turnover regulation for both 3P proteins and other subunits. So interestingly, we further found that this loss type aneuploidy might be associated with the protein thermal stability using cysteine, for which I do not have time to show today. So to summarize the studies on the protein turnover or lifetime, I showed you guys that at the bioinformatics side, we developed the ISW workflow. At the DRA method side, we devised the Boxcar Max DRA as a new DRA method. And at the application side, I showed you that we analyzed the lifetime associated mRNA alternatively splicing isoforms and protein phosphorylation and cancer and euploidy for lung cancer. This brings me to the end of my talk. So my third advice to the student would be try to attend as many conferences as possible. You see, I met Rudy in this HUPO, and I always go to SMS, staying together with our colleagues like the webinar we have together today would certainly strengthen our mutual support and help us to get more spotlight and colleagues and friends. So very importantly, I want to highlight and acknowledge the other community, the Chinese American Society for Mass Spectrometry, CASMAS. We are proud to have more than 1,500 members and 2 annual conferences, and there is another one coming in August, so please register. So to make an outlook for this research direction I discussed today, we will continue to study protein turnover and lifetime with these scenarios I mentioned to you, but also to extend this into different contexts, such as how would the protein lifetime change upon change of cellular localization, the other PTMs and their turnovers and how do they crosstalk with each other, and the protein-protein interactions, and whether, for example, whether a protein would be stabilized by a protein inhibitor. So I think this endurance in context concept is not only important for individual proteins, but also for individual young proteomic scientists like us. So we have to be persistent during our lifetime and the scientific journey, from PhD to postdoc and to PI, to work with colleagues who can stabilize us and enlightening us, and be ready to do so in the future for other people. So with this, I want to make a final brief acknowledgement. I want to thank my mentors and advisors from like Len and Mark Lemon, and the current Liu Lab members who contributed to the studies I presented today, and other members and alumni. If there is anything significant, this belongs to our team. So the KSMS members, collaborators, and the community supporters, unfortunately, I cannot read them one by one. And thank you for your attention. I'm here for your questions if you have any.
Yeah, thank you, Yansheng. That was wonderful. I also really like your spin and advice. I think this is really great advice for our young scientists. And I love that you come to US HUPO, and that's wonderful. Yeah, so yeah, I mean, I think what you showed is like the perfect example, right, of a combination of very high-end technology and then amazing application to really interesting biology. I think this is really a wonderful message. So actually, I was impressed also by your, what you called ISW scoring, right? The heavy and light pair. And you mentioned you work together with Biognosis. So have they implemented this? I know they have implemented some, but have they implemented also your features into their releases?
Yes, so upon the publication of our joint paper, they already make this possible. So it's only one click now. In SpectraNote to use ISW workflow instead of labeling workflow. So we find that this could be very useful for those pulse-like experiment with shorter labeling time course. So because, you know, once you go longer for this experiment, sometimes the cells are overgrowth and become quite inhibiting to each other, which will affect your mathematical model. So if you use a shorter time point, ISW workflow will be quite useful. Yeah.
Yeah, cool. And yeah, I mean, I particularly also like your application to PTM, of course. And I think there it's particularly interesting as they are so highly dynamic, right? And so have you looked at any other PTMs other than phosphorylation so far?
Yes, so in the lab, we are actually extending this study into different cell lines and different PTMs. I mean, of course, Bernhard Kuster's group already had a very nice paper measuring the protein acetylation and the ubiquitination, and we can learn a lot from the data and the complexity of the data. And in the lab, we are now focusing more about, you know, whether could we measure these different parameters for different cancer cells, and to understand better about how this relative change between cells would contribute to the phenotype instead of a steady state measurement on one cell line.
Interesting.
Yeah.
I have a question.
Sure, Jeff.
I'm curious, did you find any specific instances where you have like the same peptide, say that's like mono or diphosphorylated, and you could see how like the effect of a second phosphorylation or even a third phosphorylation would affect the turnover of that peptide and protein?
Yeah, so that's a great point, Jeff. So actually, we do have the multi-phosphorylated peptide in the data set, right? So the interesting part, I mean, is that we observed in general, many phosphorylation actually tend to stabilize the protein or peptide turnover, which was heavily discussed by Mihail Savatsky's group, that this is a kind of a default behavior. However, the relative difference was not discussed in the paper. So if we think about that trend, so we would expect that more phosphosite would stabilize more the protein turnover and extend the lifetime. So that's exactly what we see. So in the paper, we have the box plot looking at the lifetime change for the singly, doubly, and multiple phosphosite in the same peptide, and we do see the differences.
Very cool. Thank you.
Thank you.
Yeah, wonderful. This is thanks again, Yansheng. This was amazing that you, and also that you took the time today. And again, you know, this webinar will be on the website, so it will be visible for anybody who is interested. So thank you, Yansheng. This was wonderful. Great.
Thank you. Thank you all.